Hi, my name is Alexander Hoffman, and the title of my presentation is Single Gene Dot Plot Analysis is Effective in Predicting Cluster Assignment in Gordonia Bacteriophages. I'd like to start by giving an overview of bacteriophages, also known simply as just phages. Simply put, they're viruses that infect bacteria. There's estimated to be over 10 to the 31st individual phages worldwide. They all vary in structure, but they all have a capsid head that encases their genome, and some have a tail extending from that capsid. An image of a bacteriophage produced by Ian Lorang, a former Carroll student, can be seen in the top right of the screen with a somewhat transparent tail extending from the dark capsid. They're found virtually everywhere on Earth. They're found everywhere that bacteria are found, and they replicate themselves by using the machinery found inside bacteria. There are a variety of different methods used to classify phages, the most common being clusters and subclusters. Clusters of phages are phages that are more genetically similar to each other than any other group of phages, whereas subclusters are subdivisions of clusters and contain phages that are even more genetically similar to each other than the phages of the same cluster that are in different subclusters. Cluster and subcluster assignments are determined using a variety of techniques, including gene content analysis, average nucleotide identity, pairwise genome map comparison, and dot plot analysis. Dot plot analysis is the most common method and is what I use in the study. Classifying phages is important for efficient research. Being able to identify phages that are closely related to each other and look at differences between them that may be due to genetic differences such as tail length or infectability of bacteria is important. At the bottom is an example of a list of phages that all belong to the A15 subcluster that can be found on Phamerator, which is a program that can be used to identify phages and also compare them and look at differences in their genomes. It's been previously shown that single gene comparison is an accurate predictor of cluster assignment in Mycobacterium phages. So these would be phages that infect bacteria belonging to the genus Mycobacterium. So the goal of my project was to determine if Gordoniophages, being phages that infect bacteria from the genus Gordonia, can be divided accurately into clusters using single gene comparison. I selected two genes to use in this test, the tape measure and portal gene. The reason I selected these two is that they're both long and ubiquitous. The reason that I look for long genes is that they are able to take on a variety of mutations without losing their function. And it's also important that they're ubiquitous and found in every single phage that I looked into. Otherwise, it would be impossible to do a comparison. My hypothesis was that single gene dot plot comparison of tape measure and portal genes will accurately predict cluster and subcluster assignments in Gordonia-phages assigned by full genome analysis. Being able to quickly identify phages, individual cluster and subcluster, would be very important to the future study of bacteriophages because as of now, the main method of determining a phage's cluster and subcluster is by using full genome analysis, but this is both expensive and time consuming. It takes a long time for the reactions to progress and the reagents are fairly expensive. Another reason that this study is important is that researchers can quickly identify phages of interest of a desired cluster. So if they wanted to find phages belonging to cluster A, for example, they could have an impure mixture of phages and they wouldn't have to take the time to separate them all out individually. They could quickly identify which ones they wanted to further purify for full genome analysis by using a single gene comparison method. This study will also increase the speed will obviously increase the speed of cluster assignment, but this will also assist in other studies. One of these being looking into phage mosaicism. Phage mosaicism is a property of viruses that, and bacteriophages, especially that they can easily exchange genetic info, genes and such with each other and with their hosts, so in this case being bacteria. The picture at the bottom shows a Phamerator map that compares two phages of the same cluster but different subclusters. The shaded regions in between show regions of similarity. As you can see, these regions have moved around a little bit, showing that these phages have picked up different regions of genetic information over time. At the start of my research, I randomly selected 30 phages from the Bacteriophage database. This is a database that holds information about all known bacteriophages. I then collected FASTA files from Phamerator for each phage's full genome, portal gene, and tape measure gene. I then created 3 dot plots using a program called Gepard, one dot plot comparing every phage's full genome to each other's, their portal gene to each other's, and tape measure gene. I use this as a qualitative and visual method of assigning cluster and subcluster. For a more quantitative and exact cluster and subcluster assignments based off single gene, I compared sequences using BLAST. This is a tool that compares entire sequences with exact nucleotide alignment. So if clusters had greater than or equal, or for phages that had greater than or equal to 50% similarity between their genes, I assign them to the same cluster. For phages with 85% or greater similarity, I assign them to the same subcluster. I then generated phylogenetic trees using individual gene sequences using the maximum likelihood model in MegaX software. I use this as a comparison method to what I had already done. This method is not commonly used in assigning cluster and subcluster, but I wanted to see how effective it would be in assigning cluster and subcluster using only a single gene. So now for my results. My first dot plot that I generated included phages' entire genomes compared to one another. On the axes, the X and Y axes are the phages' individual clusters labeled. So the clusters that were represented in this study are cluster A, cluster CQ, cluster CR, cluster CS, cluster CU, cluster CZ, and cluster DE. Each of these individual clusters represent anywhere from 2 to 6 individual phages genomes. The phages on the x-axis are the exact same as the phages on the y-axis, so they're compared to one another as well as to themselves. Regions, darker shaded regions in the dot plot show regions of greater similarity. So as you can see, the black diagonal line running from the top left to the bottom right corner are phages being compared to themselves, so they have 100% similarity. As you can see, when clusters are compared to themselves, there's more dark shading showing that they have greater genetic similarity. I then generated a dot plot using FASTA files of the phage's portal gene. The phages are in the same location on the X and Y axis. So as you can see, the diagonal black line is still present when they're compared to themselves. And generally, the clusters still have the same shaded regions when compared to each other, which is a qualitative marker that the portal gene is an effective predictor of cluster assignment. I then went on to create a dot plot using the tape measure gene. Again, the phages are in the same location on the X and Y axis. And again, you can see the darker shading occurring when clusters are compared to one another. Again, indicating that the tape measure gene is an effective predictor of cluster assignment. Here are those 3 dot plots again, all shown on the same slide. So the full genome comparison on the left, in the middle is the portal gene, and on the right is the tape measure gene. As you can see by looking at these 3 dot plots together, they have similar regions of shading, which again is an indicator that both the portal gene and the tape measure gene are accurate predictors of cluster assignment. Here are 3 dot plots I generated to show similarity between phages that I assigned to the same cluster, phages I assigned to the same subcluster, and phages that I assigned to different clusters. So in the top left, the dot plot labeled A are phages assigned to the same cluster based off their portal gene, in this case Catherine G and J swag. You can see from the dark diagonal line with hardly any breaks in it that there is great similarity, genetic similarity between these two genes, so I assigned them to the same subcluster. If you go to the top right in figure B, when comparing the portal genes of Leonard and Verity, there is still some similarity with that diagonal line, but it is not as consistent, so I assigned them to the same cluster and not the same subcluster. If you look at the bottom, when comparing the tape measure genes of Club L and Madeline, there's hardly any similarities. There's just a few darker shaded points. So these phages I assigned to completely different clusters. The data in the dot plots was more qualitative, and I wanted something a little bit more quantitative. So I used BLAST to look for exact nucleotide alignments between phages genes. So phages that had greater than or equal to 50% alignment in their genes, I assigned to the same cluster, whereas phages with greater than or equal to 85% alignment, I assigned to the same subcluster. So starting with the portal gene cluster recovery rate, using BLAST, there was a 100% recovery rate, and the value in the parentheses, 1.07, was the p-value from a t-test comparing my data to a previous study showing that single gene analysis of Mycobacterium is an effective predictor of cluster assignment. Then on to the subcluster recovery rate for the portal gene, it was 93.3 plus or minus 8.9% with a p-value of 0.86. The plus or minus value is a 95% confidence interval. Onto the tape measure gene, the cluster recovery rate was 96.7 plus or minus 6.4% with a p-value of 0.9. And the subcluster recovery rate was 93.3 plus or minus 8.9% with a p-value of 0.86. Since all these p-values are greater than 0.05, this means that there was no significant difference between my data that I collected and the previous study that showed that single gene analysis is effective in predicting cluster assignment. This is one of the phylogenetic trees I generated using the MegaX software, looking at a different method of single gene comparison to assign clusters and subclusters. Phages in black were assigned to the correct subcluster. Phages in blue were assigned to the correct cluster but wrong subcluster. And phages in red were assigned to the wrong cluster entirely. For this phylogenetic tree, I used the portal gene. This had a cluster recovery rate of 96.7% and a subcluster recovery rate of 86.7%. Similarly, I generated a phylogenetic tree using the tape measure gene. As before, phages in black are assigned to this correct subcluster. In blue, the correct cluster but wrong subcluster, and in red, the wrong cluster entirely. The tape measure gene had a cluster recovery rate of 96.7% and a subcluster recovery rate of 86.7%. In conclusion, the tape measure and portal single gene analysis is an accurate predictor of cluster assignment when compared to full genome analysis in Gordonia phages. In the future, scientists should look to expand the sample size. In the study, I only used 30 bacteriophages, which is not nearly enough to be representative of the seemingly infinite amount of unique phages on Earth. They should also look to test other genes. Some candidate genes might be tail chaperone genes or tail assembly genes that seem to be ubiquitous and somewhat conserved throughout all phages. They also might look to analyze other groups of phages. An example of another group of phages that might be of interest are coli-phages that infect E. coli. And here are my citations. Thank you for listening to my presentation.